Good afternoon, everyone, and welcome to Advanced Plant Science Seminar. I'm very happy to have Dr. Brent McCallum from Agriculture and Agri-Food Canada in Morden with us here today to deliver a presentation. And I'll ask Dr. McCallum to introduce himself. He is no stranger to the department, and yet we may have some new faces here who don't have as much knowledge about who you are yet. So welcome, Brent.
Thanks, Yvonne. Yeah, welcome everyone. My name is Brent McCallum, as Yvonne said. I'm a proud graduate of this department as well, my bachelor's and my master's degrees from this department. So I always enjoy coming back here. It's always a good experience to interact with you folks and all the faculty that I still know. And we have Teddy here today too, so he's a good friend of mine as well. So today I'm going to tell you a little bit about some projects that we've been working on over the years, and they kind of all revolve around this one resistance gene, Lr34. First of all, just to acknowledge all the people that I work with on these various projects. From AFC in Morden, Colin Hebert, Tom Fetch, Maria Antonia Enriquez, Andre Belichek, Shiben, Abdul Salam Dakari, Mark Jordan, and then our AFC folks in Ottawa now, Sylvie and Frank Yu. AFC in Swift Current, Ron Knox, Richard Cuthbert, Ferdisa Bakori, and then I've got collaborators here in this department who contributed to this presentation as well. Kurt McCartney, Gary Martins, Martin Enns, and Rob Golden all played a part in this. And of course, can't get any of it done without the technical assistance that I have, really great people that I work with, Elsa, Leslie, Debbie, Winnie, Pat, and Nadine. So yeah, with that, I'll get on to the research here. Okay, so this is the disease I primarily work on, although we're going to be talking about a few weak diseases today, but wheat leaf rust is the one that I work on. It gets the name from these rusty colored spores you see here. And these spores are easily airborne, and they can travel like long distances in the upper atmosphere, thousands of miles really. And when it gets bad, you can see the kind of devastation that it can cause on a wheat crop, where it just kind of takes out the flag leaves and really causes a lot of damage. So yield losses can be in the range of 3 to 20%, but currently we're doing a really good job of controlling leaf rust with fungicide resistance and fungicide application and genetic resistance. So how does the rust get here? Well, this pathogen does not overwinter here like some of our pathogens can. It has to be blown in from the southern US and then the northern US. So it starts down here on winter wheat, and then as the crop starts to mature, it moves further north, and then spring wheat starts to mature and it comes up here and then into our area here. So every year you get a south to north migration, and it goes kind of this way into our area, but then it comes over here into southern Ontario and Quebec. And then on the western side of the Rockies, there's another route into Alberta and BC. Okay, I'm going to talk mostly about resistance genes and controlling leaf rust. And probably the most famous guy in this whole research area is Peter Dick. And that's a picture of him here, and he's from Burwaldy, Manitoba. So Burwaldy's, when you go to Morden, Winkler, there's an area, there's the Winkler Bible Camp that you pass by. That's Burwaldy. So probably the most famous person in the world is from our local area in this research area. So of the 82 officially named leaf rust genes, Peter discovered in whole or in part 21 of these genes, and that's a list there. But I'm going to be talking today mostly about Lr34 and Lr67, because these genes are a little bit different, a little bit better than the others. And he also developed the Thatcher near isogenic series. So almost all the studies that I'm going to talk about today use these lines, but everybody working in the world today on leaf rust uses those lines as well. So he's done some great contributions. Okay, the first type of gene I'm going to talk about are race-specific resistance genes. So they are effective only against a certain biotype of wheat leaf rust we call a race, but they're also only effective against wheat leaf rust. So they don't do anything against stem rust or stripe rust. They only control wheat leaf rust. And such a gene is Lr2C here. So what we have here is Thatcher, which is a universal susceptible cultivar. So you can see the big pustules on it produced by this isolate here, 198-1. But when Peter created this near isogenic line, so it's 99% genetically similar to Thatcher, but it has the addition of Lr2C. So these two lines are very genetically similar, but this one has Lr2C. And you can see when you inoculate with this isolate that it gives good resistance. So just small pustules, so very little sporulation. So we're getting great resistance, and that would work very well in the field. However, the pathogen can change, and genetically it can overcome this resistance in this line. And that's what we see here with a different isolate, 196-2. This one is changed genetically, so now it's able to overcome Lr2C, and you get back to these big pustules, susceptible pustules. So this is nearly all our rust resistance genes are race specific, and they work just like this one. However, we have a few that are not. And that's what I'm going to talk about today, are these race non-specific durable resistance genes. And the classic one is Lr34, as Peter discovered this gene. Slow rusting adult plant resistance. So it's great for leaf rust, but it also controls stem rust, stripe rust, powdery mildew, and FHB. So all those other race-specific resistance genes don't do that. They only control one pathogen, whereas this one controls many. And it synergistically combines with other leaf rust, stem and stripe rust resistance genes, and I'll show you how it does that. So this gene is not race-specific. We've never seen an isolate that's more virulent on it. So it has the same effect on all isolates and provides durable resistance, so over a large acreage and many years. And this one has been cloned, and it codes for an ABC transporter. So it's, again, different from almost all the other known resistance genes. Those are based on recognition of the pathogen. This one's not based on recognition. It's a transporter, and it moves something within the cell. We don't exactly know what that is, but that's what it does. And then we have two other genes, Lr67 and Lr46, and I'm going to talk about Lr67 a little bit, but they're functionally very similar to Lr34. Okay, so just to have a look what it looks like here, it's an adult plant resistance, so you have to test it on adult plants, and these are flag leaves. So here's Thatcher again, our susceptible, and here's Thatcher Lr34, again a near isogenic line, only one gene difference from Thatcher. But you can see it produces much fewer pustules. They're a little bit bigger down at the base here, but then they get smaller towards the tip. And you'll also see this big leaf tip necrosis characteristic. And that's not necessarily great. That's not really what we want in our wheat, is for the tips to die. But it is a phenotypic marker that this gene also produces. Okay, does breeding for resistance work? You know, we've been at it for 100 years, so it better work, right? And this was a study that we did with Gary Martins here at the University of Manitoba. Gary tested 45 historic wheat cultivars at our Carmen Research Farm. And so these were released from, this is the year of release of these cultivars along the bottom here. So from even before 1900 right up until 2000. And you can see that the ones that are older here are pretty susceptible. This is the amount of leaf rust they're getting on the flag leaves. So these are getting a lot of leaf rust. But as we move to the newer ones, we're getting less and less. And then the very new ones, we're not getting any leaf rust at all. So that's a goal of breeding for resistance, right? You want to control it, and that's what we're doing. Okay, so when we breed for resistance, what we're doing is introducing resistance genes into our cultivars. But which resistance genes? You know, what are we putting in there? So the first one we incorporated was Lr16. And this is a plot that just shows you for the whole CWRS seeded acreage, which is the biggest wheat class, what's the proportion of Lr16 in that seeded acreage? So you can see before 1950s, there really was zero of that gene. But then here we started to see a cultivar called Selkirk. So in Selkirk, it had Lr16. So it went up to about 30% of the crop. But then after Selkirk, it went down. It came back up with cultivars like AC Berry. Right now, it's back down a little bit, but it's still a very effective resistance gene. Another one that's race-specific, Lr2A. I showed you Lr2C. This is just an allele of Lr2. It's Lr2A, very similar. And before the year about 2002, there wasn't any of this gene in our crop. But after that year, it started to go up, and now it's gone dramatically up. It's not necessarily that it's the world's greatest gene, but sometimes these genes are in cultivars that do very well, right? Just by chance. And that's sort of the case here because we have some really big cultivars lately like AAC Brandon, that's the predominant one that has Lr2A, so that drives that up. And then Lr34. So these three genes are kind of the biggest ones that we have in our Canadian wheat. And here, okay, before 1973, there was really no Lr34 in our crop. And then with the release of Glen Lee from the University of Manitoba, in 1974, we started to see Lr34 kind of at low levels because Glen Lee was never a huge cultivar. But then we started to see other ones with Lr34, like Roblin and other cultivars like that. And so then it dipped a little bit in the early 2000s, but now it's gone up even higher. So right now it's sitting at somewhere over 50% of our current wheat cultivar has Lr34, with current wheat seeded acreage, we'll say. Okay, so I said Lr34 is a durable resistance gene, so what do we mean by that? That means it's been out in production over large acreage for a long period of time, and it hasn't broken down to the pathogen. There's no pathogen that's virulent on it. So these are the kind of annual leaf rust scores in our leaf rust nursery for two lines. The blue one is Thatcher Lr21, again, a near isogenic line with Thatcher with the gene Lr21. And then the red one is Lr34. So you can see there's a lot of environmental variability each year because it's cooler, it's warmer, we have more rust, less rust. So you get a little bit of bouncing around here. But even if you went back another 10 years this way, these two would be pretty low, 20, 30, 40 in this range. But then in the year 2012, we saw a change in the pathogen population. And now Lr21 is not very effective anymore because the pathogen has changed. It's overcome resistance in Lr21. And so now we're seeing a lot of leaf rust on Lr21. And then it depends what's in the population, but it's stayed up pretty high since that time. So we would say that that's a race-specific gene. With race has changed, the pathogen has changed, and it's been overcome. But Lr34 keeps ticking along here, and it probably will forever. It's always nice and low. So that's what we talk about when we mean durable resistance gene. Okay, I was going to talk about how it combines well with other resistance genes. So this is again in that same nursery where we have our leaf rust nursery we conduct on an annual basis. Here's Thatcher up here, so it's pretty as susceptible as you can get. Thatcher 34 is the red one, again bouncing up and down kind of between the 40 and 0 here. And another line, Thatcher Lr33, is in the similar range, somewhere 30, 40, 20 in that range. So when you look at these two lines, you say, okay, well, they're kind of intermediate resistance, but what happens when you put them both together, right? So we have a line that has both 33 and 34, but it's still a Thatcher isoline. And that one is this green line down at the bottom here. So 1 + 1 does not necessarily make 2. It's more like a synergistic effect in that you get even better resistance with combinations of resistance genes when you have Lr34 involved. And we can track these in populations as well. So these are double haploid populations that Colin Hebert generated. And The first one here is Thatcher Lr34 by Thatcher Lr32. And these bars represent 20 or 30 DH lines within a segregating population that have been scored for these genes. So this would be the average for all the 20 or 30 DH lines that had neither gene. This is the average for the ones that had Lr34. So you can see it's about 30%. The average for the ones that had 32 here, it's about 50%. So it's bringing it down a little bit, but not as much as Lr34. But you look at the ones with both genes, and now you're getting a really serious reduction, right? So the two are working much better. And down here, it's another DH population, but instead of Lr32 as a second parent, we have Lr13. And we look at the lines with Lr13 alone, they look basically the same as Thatcher's. So you'd say, well, this gene's not really doing anything, right? But then if you put it together with Lr34, these lines here now, we get a reduction from Lr34. So the Lr34 is doing something with Lr13, even though Lr13 is not that effective on its own. So it has some really interesting effects. Okay, I said before there's other genes that are similar to Lr34, and they're Lr67 and Lr46. So Lr67 was also discovered by Peter Dick, but it was mapped to chromosome and characterized and named by Colin Hebert in 2010. So it's like Lr34 in that it's a multi-pest resistance gene, conferring resistance to leaf rust, stem rust, and stripe rust. And it has also been cloned, and it also codes for a transporter. This time it's a sugar transporter, so it also transports something. So it's not involved in recognition, it's a transport protein. So we wanted to look at some of the phenotypic effects of Lr67. Is it similar to Lr34 in the way that Lr34 interacts with other genes? So this was a double haploid population that Colin created with Thatcher Lr67 and Thatcher Lr16. So the double haploid lines with neither gene were susceptible, just like Thatcher. The lines with 16 were also pretty darn susceptible, up here at nearly the same rating as Thatcher. 67, the lines with Lr67, you can see they're up here at about 55%. So if you remember Lr34, it was down here at about 30, right? So 67 is not as effective on its own as Lr34, but you put it together with 16 here and you get the same effect as you do with Lr34, that you get a combination effect of the two genes. So Lr67 seems to work like Lr34 in that it combines synergistically with other genes. So I said that Lr34 is a multi-pest resistance gene. We've talked about leaf rust so far, so now we're going to branch out into some other diseases. So this is stem rust. It's another disease, but caused by a different pathogen. And both these pictures are pairs of near isogenic sister lines. This time they're not in the Thatcher background, they're in the SUMI3 background. SUMI3 is the highly FHB resistant line. So in both cases, the line on the left has Lr34, and the line on the right has a susceptible allele of Lr34. And one thing with stem rust, you can see all this lodging that's going on here. The stems are breaking, and so you're getting no yield out of this plot at all because the heads are on the ground or they're falling off. Whereas this one here is still standing up. So the resistance gene has provided enough resistance so that you get at least a crop. And then here too, again, standing up with the ones Lr34, falling down the ones without. So yeah, it's a stem rust resistance gene as well. And then this is work from Colin Hebert. It's a little more complicated, this concept, but it acts also as a counter suppressor of resistance. What do I mean by that? Well, Thatcher contains a resistance gene that is effective against the stem rust race QTH, but it's suppressed by another suppressor gene in Thatcher. So when you look at QTH down here, this is just a susceptible line here. This one is Thatcher. So even though Thatcher has a resistance gene that should be effective against QTH, there's another gene, a second gene that's suppressing the effect of that first resistance gene, right, in Thatcher. However, if you take Thatcher and introduce Lr34, it still has the resistance gene, it still has the suppressor, but now it has Lr34. Now the Lr34 acts to counteract the suppressor, and this resistance gene is able to express. So it's kind of a 3-gene interaction here. You've got the resistance gene, the suppressor gene, and Lr34. If you don't have Lr34, the suppressor suppresses the resistance gene, it's not expressed. If you do have Lr34, it knocks out the suppressor, the resistance gene is able to express. So it's got some interesting qualities. Okay, one interesting thing that I was researching was the seedling resistance of Lr34, because we talk about Lr34, Lr67, Lr46 being adult plant resistance genes. But can we detect it at seedling? Well, the typical greenhouse temperature is 15 to 28 C. Lr34 cannot be reliably identified at the seedling stage. Dick reported that seedling expression was variable, I guess depending on temperature. Rubales and Nix in 1995 demonstrate Lr34 had increased latent period in seedlings at 8 degrees C. And Agarwal in 2003 reported seedling expression of Lr34 at 14 degrees C. So we set up this experiment with our double haploid population of Thatcher, crossed with Thatcher Lr34, again near isogenic lines. And this time we grew the plants at 8 degrees, so a really cold temperature. And then we inoculated them with leaf rust. And Thatcher, you'll see it's not the clearest picture, but it has susceptible pustules on it. Whereas Thatcher 34 has smaller pustules, a resistant response. And you can also see this leaf tip necrosis, which is, you never see this in seedlings at normal temperature. You always see it at adult at normal temperature. But if you turn the temperature down to 8 degrees C, now you see it at seedling, right? And then if you look at the DH population, those lines that didn't have Lr34 or had the susceptible allele of Lr34, they had big pustules and no leaf tip necrosis like these ones, this one here, these two here, yeah. Oh, sorry, I'm looking for the minus signs, these two here and this one. So these guys, they have big pustules, no leaf tip necrosis. But the ones that do have Lr34, the resistance allele of Lr34, they have small pustules and they have this leaf tip necrosis. The two things go together, right? Where you see leaf tip necrosis, you're going to see small pustules. So right throughout the population, those two phenotypes co-segregated. So every time we saw leaf tip necrosis, we had effective seedling resistance, and where we had the susceptible allele, we saw neither trait. Okay, so that's interesting, but what happens if you take these plants and grow them in those cold conditions for much longer? So I tried that, and I grew them for 7 months, because it takes forever for plants to grow when it's that cold, right? So what do they look like after 7 months in 10 degrees C? Well, Thatcher doesn't look too terrible. It does head eventually, even at 8 degrees C. Spikes are not fantastic, so it's not the happiest plant, but at least it's looking like a wheat plant. Thatcher Lr34, on the other hand, doesn't look very good at all. So it's got huge leaf tip necrosis on the flag leaves and on other leaves. And in this case, the whole spikelet was aborted, right? So it really has some detrimental effects. These two are only different by one resistance gene. So that's the effect of this gene. And then when you look at the progeny from the double haploid populations, generally the ones with a susceptible allele at Lr34 looked fairly normal. The ones with the resistance gene always had this huge leaf tip necrosis, and quite a few of them had these aborted spikes. And this is also true in different genetic backgrounds. So Glenn Lee, we talked about here, the one from U of M that has Lr34. Again, it has big leaf tip necrosis, but it was able to form a spike at least. So this is a good resistance gene, but it's not good in all situations, right? This is a negative trait that it has. So Kurt and I were talking about that and saying, well, in winter wheat, it'll often grow under these conditions, 8 degrees, 10 degrees, those kind of things when it's coming out of the spring. And we don't have a lot of Lr34 in winter wheat. And this may be why. If your plants look like this, you're not going to keep them, right? You're going to throw them out. Okay, so one of the things we've been doing lately is the effect of Lr34 and Lr67 on Fusarium head blight, because both genes have a positive effect on controlling many wheat diseases caused by biotrophs. So leaf rust, stem rust, stripe rust, powdery mildew, etc. But would they have an effect on a hemibiotrope like Fusarium graminearum? So again, we used our 2 double haploid populations that segregated either for Lr34 or Lr67, and they were tested in 5 Fusarium graminearum inoculated disease nurseries over 4 years. So all the lines were rated for visual symptoms, and then harvested grain samples were tested for DON. So these are the results that we saw. This is DON content for these 92 double haploid progeny lines from the cross of Thatcher Lr34 by Thatcher 13. In this case, we did use 13 rather than just Thatcher, but Lr13 had no effect on FHB, so we're just going to ignore that one for these purposes. So the blue bars represent the average of the lines with a susceptible allele of Lr34, and the red bars represent the average of the lines with the resistant allele of Lr34. And then this indicates significance. So in 2016, this is in Morden, these are all in Morden, there was a significant effect. The lines with Lr34 had significantly less DON than the ones with the susceptible allele. We also saw that in 2017, but not in 2018 or in 2022 in Carmen, but we did see it in 2022 in Morden. And overall, there was a significant effect of lowering DON caused by Lr34. So this is pretty interesting because, say, Lr34 is in about half of our wheat varieties. One of our major objectives for wheat breeding is FHB resistance, right? So knowing that Lr34 actually conditions FHB resistance is really helpful. Okay, so what do the lines look like here? So this is just a histogram of these 92 double haploid lines from that cross that we were just talking about, Lr34 by Thatcher 13. And you can see that they don't all fall into one discrete category of DON here, but the ones with the susceptible allele tend to be higher. There's sort of a normal curve with both sets of lines, but the curve is shifted a little bit to the right for the ones with the susceptible allele and a little to the left for the ones with the resistant allele. Now, that was Lr34. How about Lr67? If these two genes function similar, does Lr67 also have an effect on FHB? Okay, so this time we had a DH population, Thatcher 67 by Thatcher 13. And again, we can ignore 13 here. So the blue bars are the ones with the susceptible allele at Lr67, and the red bars have the resistant allele at Lr67. And sure enough, for nearly every environment, the lines with the resistant allele at Lr67 were had less DON accumulation than the ones with the susceptible allele. And overall, that was a significant effect. So again, Lr67, a different gene from Lr34, is functioning very similar to Lr34. And again, when we look at the histogram of the lines here, you can see it's not, because this is a population, we don't get all the lines being the same. There's two normal curves, but the susceptible ones are shifted to the right and the resistant ones are shifted to the left. So that's good news. Okay. Now, I talked about cloning of these genes. It's not my area of expertise, but both Lr34 and Lr67 have been cloned. So Lr34 encodes for ABC transporter, and Lr67 encodes for a hexose transporter, so a sugar transporter. But it's interesting, they're both code for transporters, and that's very different from every other resistance gene that codes for recognition. So these genes are not only different from other known R genes, but they encode proteins of different classes. Okay, so this was a study done by Salam Dakouri in 2014, and he analyzed a worldwide collection of 310 spring wheat accessions both for their leaf rust phenotype in the field, but also for their Lr34 haplotype. So the haplotype consists of 4 or 5 different locations, but these 2 that are highlighted here are in the coding region of the gene. So there's an indel 11 and a SNP at 12. So the resistant haplotypes here that have the resistance phenotype in the field all have a deletion at indel 12, and they have a C at SNP. Indel 11 a deletion and C at SNP 12. Whereas all the susceptible haplotypes have an insertion at indel 11, TTC, and they have T for SNP 12. That was the dogma, that there's only those 2 types out there. However, Sylvie found 2 lines in that collection that have a recombinant haplotype. So they have the null allele for indel 11 and they have a T for SNP 12. So what do those cultivars look like? Are they resistant or are they susceptible, right? So Sylvie Cloutier set up this series of crosses here with those 2 cultivars. I'm just going to call them O and K because I have trouble pronouncing those words. Okay, so O was crossed with Thatcher. So this is the recombinant haplotype crossed by the susceptible haplotype. And she also crossed the O line with Thatcher Lr34, so the recombinant type with the resistant haplotype. And then she took those 2 populations and she combined them. So now we had 8 lines that had the resistant haplotype, 12 lines that had the susceptible haplotype, and 33 lines that had this recombinant haplotype. She did exactly the same thing to the K line and ended up with similar numbers of lines with the 3 haplotypes. So then we tested those 2 populations over 5 years for leaf rust resistance to see what the effects of the different haplotypes were. So that's what you see here at the top. This is for the O line, and all the progeny with the resistant, or the susceptible haplotype, the susceptible haplotype is the insertion with T. They were all fairly susceptible, but again, a range because of the population, right? The ones with the resistant haplotype were all resistant, but the ones with the recombinant haplotype were definitely intermediate, a bigger range and in the middle. And the same thing for the T line crosses. The resistant haplotype was down here, resistant haplotype was resistant, susceptible haplotype, susceptible, and the recombinant haplotype right in the middle. So then when you look at the statistics on this, you have 3 classes in both populations. The susceptible haplotype is most susceptible, the resistant haplotype is very resistant, and the recombinant haplotype is right in the middle. Then when you took a look at the effect of the indel, whether it was the null or TTC, that had a significant effect on leaf rust in both populations. And the exon, the SNP12, also had a significant effect on leaf rust in both populations. So it tells you that even though there's 2 differences, those 2 differences are additive, they're both important, and it's an additive effect between 1 and 2 to get you to the resistance. Okay, So just to summarize things then, breeding for resistance to leaf rust and other wheat diseases has produced excellent results in Canada. So Lr34 is key to these efforts and provides durable, effective resistance to wheat leaf rust and other diseases. So Lr34 and Lr67 combine in a synergistic manner with other resistance genes. At low temperatures, Lr34 is harmful to plant development, so it's not all good news. Lr34 and Lr67 reduce visual symptoms of FHB and DON in harvested grain samples. So that's probably our most significant finding lately, is that it has a significant effect on FHB. And both the exon 11 indel and the exon 12 SNP are additive for the functioning of Lr34. So that's my story on those genes, and thanks for your attention, and I'd be happy to take any questions you might have. Yeah.
Thank you for the great presentation. It's very much informative. I'm just curious, my question may be silly to ask, because Lr34 and Lr67 are so great, both are resonance specific, and does it make sense at all to say pilonate the two of them and not the hybrid?
Yeah, that's a great question, actually. Something we discuss a lot between us, Kurt and Colin and myself. There's actually the three genes. I didn't discuss Lr46 very much, but even though we haven't done the studies on Lr46, it seems to work very much like Lr67 and Lr34. So the three genes, if you combine them all together, would you get a greater effect? And I say yes, and Colin says no. So it's a good question. But we did do a study on the cultivar Carberry, which has both Lr34 and Lr46. And in that population, we did a double haploid population between Carberry and Thatcher. So we had all the lines distinguished by all the genes they carried. So the class of genes, or the class of lines with both Lr34 and Lr46 were significantly lower than the ones with either Lr34 or Lr46. So that seems to prove that, yes, it helps to combine the genes. And they both also combine very well with all the other genes that were in that population. So we're learning more about 46 as we go, because I didn't put it on the graph here, but we know it's in some of our Canadian varieties. We just don't know what the percentage is, because the molecular markers for Lr46 are not as good as the ones we have for Lr34 and Lr67. So we're not confident in scoring our cultivars for 46, but it's in there. And it's definitely having a good effect. And well, who knows? Maybe it also has an effect on FHB, which would be a very positive effect for Canadian wheat, for sure. So yeah, very good question.
Yeah. So you named Lr67 and 34 as multi-pest resistance.
Right.
Do you mean just within the fungal pathogen species, or can they compare also resistance against bacterial?
Well, bacteria is a good question. I don't know if anybody's tried these genes against bacteria, but they do work against viruses, like BYDV. Lr34 is actually named BYDV1, like it's the first resistance gene against BYDV in wheat. So it works against virus infection, but does it work against bacteria? I'm not sure. It's a good question, because right now there's that bacterial leaf streak is a big problem in wheat. And do these genes help control that disease? I don't know, but that'd probably be a good research project to find out.
And the other question that I have is, I think in the greenhouse experiment, you grow the plants at a lower temperature. If I'm correct, if I understand correctly.
That one experiment where we were doing the low temperature test.
And the lines that carries an Lr34, they are doing well, I think, at lower temperature, is that right?
Well, like at low temperature, they get the leaf tip necrosis, like even on seedling leaves. And yeah, if you keep it at low temperature for long enough, Lr34 basically kills the plants or gets close to killing the plants, right? It's really detrimental. So Kurt and I were talking about winter wheat, and Lr34 is not very common in winter wheat, and that could be the reason why. Because at cold temperatures, your winter wheat's trying to grow in the spring or in the fall when temperatures are 10 degrees or 8 degrees. And if Lr34 is having that kind of detrimental effect, you really wouldn't want it in your line. So, although, like in the spring wheat, it seems to be great because I didn't talk about yield here, but we're seeing the proportion of the crop with Lr34 going up. And at the same time, we're seeing the yield of those cultivars going way up too. So definitely it doesn't hurt yield significantly and helps on all kinds of disease resistance in spring wheat. May not be the case in winter wheat.
The other question that I have is you showed us a list of LR genes, I think in one of your slides, at the very beginning. Only the two are cloned, that 34 and 67 are the only ones that are cloned and cut?
No. Those are the two, there's the 3 race non-specific resistance genes. And yeah, 2 of them have been cloned, 34 and 67. But then there's all the, the other 76 race-specific resistance genes. And of those, probably 10 have been cloned now. So yeah, more every day. But their sequence is all like these leucine-rich repeat at MB, LRR genes that are involved in recognition of the pathogen. So all those are recognition genes. These ones are transporter genes, so that's what makes them different. But how they work is still a good question. I haven't seen any good papers out there because the susceptible allele of Lr34 is also a functional transporter, right? So you got resistant allele as a functional transporter, susceptible allele as a functional transporter, but somehow those 2 changes that I showed you, the one indel and the one SNP, makes the resistant allele different enough to cause all these phenotypic effects, right? So just how is a good question. Sure. Yeah.
Did those 2 lines that Sylvia discovered, they have leaf tip necrosis as well for Lr34?
That's a good question. We did those all in the field. I think they do have a little bit of leaf tip necrosis, but yeah, it's a good question. I don't know. It'd be interesting to try those actually in that low temperature test and just compare the 3 alleles for leaf tip necrosis. Because at the low temperature, you really see that phenotype stand out. Yeah.
So leaf tip necrosis in fields trials is kind of notoriously, sometimes it's there, sometimes it's not. Is it temperature? Is it that driver? Why that's happening?
A lot of people think that it tends to be more effective at cold temperatures, and you will also see more leaf tip necrosis at colder temperatures, right? So, and then you get extreme and take it down to 8 degrees, and then you'll see a ton of it, right? But that's what they say in the field too. If you have cooler nighttime temperatures, in the 15 degree, 10 degree area, then you'll see better expression of Lr34, better disease control, where you're getting down to the 0 leaf rust, and you'll also see more leaf tip necrosis. Whereas if it's in the high 20s, now Lr34 can sometimes get up to 50 or 60% severity, and probably very little or no leaf tip necrosis. Yeah, so temperature does play a part for sure. Yeah.
Maybe this is a bit more naive on my part. You had done that experiment with the stem rust where you found that Lr34 gene enhanced the resistance and counteracted the suppressor gene.
Right, yeah.
Do you know about stacking the different rust resistance genes, whether it's SR, LR, or the stripe rust ones, and they work in conjunction to decrease the severity of say stem rust or leaf rust or stripe rust, permitting them in the area?
Yeah, that's a great question, and that's something we've worked on. We had this experiment with Pasqua that had 5 different resistance genes, and you could definitely see the 4 resistance genes other than Lr34, when you put them all together, it still wasn't really very resistant. You know, that those lines would get to about 20% or so. But if you put Lr34 together with any of the other 3, you would get a pretty good resistance. And then if you put Lr34 together with 2 of the other 3, it's even better. And then if you put all the genes together, so that's Lr34 with the 3 others, then it's pretty much completely resistant. And then that Carberry experiment I was mentioning too, it just happened that Carberry has like 7 resistance genes. And so when we did the double haploid populations, you could segregate them out into the different categories that had, you know, this gene, that gene, this gene, that gene, and then all the combinations. And definitely the combinations of genes, when they involved 34 or 46, were better than everything else. Yeah, so gene combinations is the way to go, for sure. And now, you know, with the frequency of all these resistance genes in our populations, breeders are making combinations that they're not even aware of, right? It's just that one parent might have 3 genes, the other might have 4. They might have 1 gene in common, so they have the potential of getting, you know, 6 resistance genes in their new lines. And when you select for the best level of resistance, oftentimes that's what you get. You know, you get a lot of genes. So yeah, unwittingly, we're definitely creating these stacks for sure. And that's good because they work well. So yeah.
I have a question. I really appreciated the timeline narrative to a lot of the early part of your presentation. You talked about shifts in races, and I wonder, could you put that in context? Like, how important is rust right now, and those shifts? How threatened are we with the resistance we have versus these shifts that you're seeing and probably studying? You didn't talk much about the change in the pathogen. You're talking about the resistance rates.
Yeah, you know, that's a really good question because early on, like in the 60s or 70s, our resistance that we introduced was usually just 1 gene, right? Because that's all that the breeders could put in at the time because they went from susceptible, crossed it with a resistant one, they got 1 gene, right? So that 1 gene would work for like 5 or 6 years, it would look great, and then the pathogen would change and boom, you know, that gene would be gone. But now, because we're getting so many more resistance genes in our cultivars, these stacks that we talked about, oftentimes, you know, the pathogen changing to virulence is not going to kill a cultivar because it has backup genes. You know, like Lr21 is a good example. When we saw that the races changed to become virulent on Lr21 in about 2012, there were some US cultivars that only had Lr21. So they became susceptible overnight, right? But many of our Canadian cultivars had Lr21, but they also had Lr34, Lr16, you know, some of these other genes. So they became a little bit more susceptible because they lost Lr21, but they still had good resistance because of those other genes. So that's where these pyramids are really, really powerful because you can lose effectiveness against 1 or 2 genes, but losing effectiveness with 6 at a time is unusual, especially if it's Lr34 or 67, because those ones have never been overcome. So yeah, good question though. It used to be much more important than it is now.
You had a really long list of resistance genes, so does that mean we have a long list that we can run through before we're really in trouble in Western Canada?
Well, yes and no. There is a lot of genes we could use. Many of those, like out of the 80, have already been overcome by the pathogen. Maybe half of them or so, but there's still a lot we could use. But because the stacks are really pretty good right now, like it's not a high priority for wheat breeders. I mean, they'll put them in if they have markers for the genes, right? So if you have Lr34, it's got a great marker. Lr21's got a marker. Lr16's got a marker. So those are kind of easy ones to put in. But after that, you know, all your lines are resistant, you know, so which ones have 2 genes, which ones have 5 genes, which ones have 4, unless you have markers, you can't tell. And then they're working on fusarium, and they're working on quality, and they're working on yield, right? So they're definitely concerned about leaf rust, but it's not the highest priority. They just want to keep what they have right now. Yeah.
That's a great place to be.
Oh yeah, no, we're in a good place. And there's fungicides too, and they don't necessarily need to put on fungicides for leaf rust. Like If it was only leaf rust, we would need no fungicide at all. But it's because FHB is not completely controlled that we do need the fungicide. But it also controls leaf rust. So, yeah.
Well, let's end on a good note today. Thank you. Join me in thanking Dr. Brent McCallum for this great presentation today. Thank you for coming.
Yeah, you're welcome. Thanks for your attention.